Hello students, welcome to EPG Pathsala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. Today I am going to talk about introduction of electron transport system from the paper Thermodynamics of Living System and Bioenergetics. The objective of this module is to understand the structure of different electron carrier that is present in electron transport system. This module has been divided into following section. The first one, we will have introduction of different electron transport system. Then we will discuss about structure of mitochondria. Then we will discuss about the different electron transport carriers such as ubiquinone, NADH and other electron carrier that is found in the electron transport system. And in the end we will discuss about different complexes that is found in the mitochondrial electron transport system. So first of all, as you know that ATP synthesis is very important for the survival of the organism. No organism can survive in absence of ATP. And you also know that ATP synthesis takes place in mitochondrial inner membrane, bacterial membrane. At the same time, it also synthesizes in the chloroplast thylakoid membrane. Electron transport system, that is very important in the energy production or ATP synthesis in bacteria, mitochondria and thylakoid chloroplast. So, electron transport system is a significant component of understanding the ATP synthesis in these three systems. Energy production takes place by three mechanisms. The first mechanism is substrate level ATP synthesis, where the substrate is involved in the ATP synthesis. The second one is oxidative phosphorylation, where the ATP synthesis takes place in presence of oxygen. The third one is photophosphorylation, where the ATP synthesis takes place in the presence of light. Phosphorylation is important because when we are producing ATP, that is produced from ADP. So, ATP combines with the phosphate and producing ATP. So, therefore, we are saying ATP synthesis as a phosphorylation step. So, in the substrate level phosphorylation, we does not require any electron transport system. But when we are talking about oxidative phosphorylation and photophosphorylation, we requires electron transport system. Substrate level phosphorylation takes place in glycolysis, where the phosphate is donated by substrate of glycolysis to the ADP, and then we will finally produce ATPs. Electron transport system is required in oxidative phosphorylation, that is taking place in the mitochondrial membrane, as well as photophosphorylation, that is taking place in the chloroplast. When you are talking about electron transport system has a number of mobile and non-mobile carriers. When you are talking about mobile carrier, that means they can move from one part of mitochondrial inner membrane to other part of the mitochondrial inner membrane or in the chloroplast thylakoid membrane. When we are talking about non-mobile electron carrier, that means they will not move in the mitochondrial membrane as well as in chloroplast thylakoid membrane. One term that is always used to define the energy producing membrane, and that is known as energy transducing membrane. So, mitochondrial membrane as well as thylakoid membrane is also known as energy transducing membranes. Therefore, it is very important to understand, to discuss what is electron transport system, and it is also important to discuss what are the different mobile electron carrier or non-mobile electron carrier present into the mitochondrial inner membrane that is considered as a model to discuss electron transport system in this module. Before going into the detail of electron transport basic, it is important to understand the structure of mitochondria. So mitochondria is also known as powerhouse of the cell because they are responsible for production of ATP, that is chemical energy of the cell. Mitochondria consist of two membranes, as you can see in the picture, is outer membrane and inner membrane. Intermembrane space, it is a space between two membranes, and then matrix. So basically matrix is separated by mitochondrial inner membrane from the periplasmic space. It also have ribosomes, it have its own DNA, that's why it is semi-autonomous. So inner mitochondrial membrane have different complexes that is responsible for ATP synthesis. Those complexes are complex first, second, third, fourth, fifth. But if you see the orientation of the complexes in the inner membrane, so all the complexes are arranged in such a way that proton transfer take place from matrix to intermembrane space except the complex 5, which arrange in such a way that it will transfer proton from intermembrane space to back into the matrix. Inner mitochondrial membrane also have electron carriers. These electron carriers are very important for the transfer of electron between two complexes. There are different electron carrier that is found in electron transport system of mitochondria. They includes ubiquinone, NADH, FADH2, cytochrome, and iron sulfur centers. Ubiquinone are two electron carriers, while cytochromes, iron sulfur center, are one electron carrier. At the same time, NADH and FADH2, which is itself is an electron donor, they are also two electron carrier. So the first electron carrier that is very important carrier, that is ubiquinone. So ubiquinone exists in three redox form. Redox form are one that is interconverted by accepting or donating the electrons. So the first form of ubiquinone is fully oxidized form, that is known as ubiquinone. The second form is semiquinone. As I already told that ubiquinone is a carrier of two electrons. So if one electron is taken by ubiquinone, it is converted into semiquinone. If the second electron is also taken by semiquinone, then it is converted into fully reduced ubiquinone. That means if you want to convert fully oxidized ubiquinone into fully reduced ubiquinone, you need to have two electrons. That can come either from NADH, either from FADH2, or any other sources. Ubiquinone is completely oxidized or completely reduced within the mitochondrial inner membrane. Therefore, it is a very important carrier of electron in the electron transport chain. You can see in the structure where we have isoprenoid unit. So the number of isoprenoid unit that is present in the ubiquinone is differ in different organism, but they always have similar type of structure. The another electron carrier is iron sulfur protein. As the name suggests, it have iron, it have sulfur, and it is protein, so it also have amino acid. So iron sulfur proteins are metalloprotein that carries iron ion bridged by sulfide ion and coordinated with the 4 cysteine ligands or 2 cysteine ligand and 2 histidine ligand. in the case of risky iron centers, which is found in the complex third. It is important to note here that we have sulfur as well as sulfide ion. So, this sulfide ion is additional to the cysteine sulfurs that is present in the sulfhydryl groups, and iron sulfur centers or iron sulfur proteins are one electron carrier based on the number of iron and sulfur atom present in the iron sulfur center. It is either 2Fe2S center where we have 2 iron, 2 sulfur, 4 cysteine and remaining that protein part, 3Fe3S center where we have 3 sulfur, 3 iron and 4 cysteine and remaining protein part, 4Fe4S where we have 4 sulfur, 4 iron and 4 cysteine and remaining protein part. Here you can see in how iron and sulfur atom are associated with each other in 2Fe2S iron sulfur center, and how the 4 sulfur that is coming from the sulfhydryl group of cysteine is also involved in the reaction. It is important to note that when we have iron sulfur center, or in other words you can say risky iron sulfur center, then we don't have 2 sulfur because we have histidine there. So, we have 2 sulfhydryl group coming from the cysteine and 2 nitrogen that is coming from the histidine. Because of difference in the structure of risky iron sulfur center, they will have different redox potential as compared to the other iron sulfur centers. Other electron carriers that is found in the mitochondrial inner membrane are cytochromes. Cytochrome contains heme protein which is responsible for making the stability of the structure of the cytochrome, and they also have iron which is responsible for carrying the electrons. So, iron which is found in the cytochromes, they exist in 2 forms, Fe3 form and Fe2 form. As you know that electron have negative charge. So, if electron is accepted by iron, it is converted into +2 form, but if electron is donated by the cytochrome, then Fe2 plus is converted into Fe3 plus. So, in this way, by converting Fe2 plus into Fe3 plus, that cytochrome is going to carry 1 electron. Similarly, NADH and FADH both can accept 2 electron and converted into NADH plus H plus and FADH2. Ubiquinone, NADH and FADH2 as well as FMNH2 are 2 electron carrier, while cytochrome B, cytochrome C, cytochrome A, cytochrome A3, these all are electron carriers which is found in the electron transport system, are 1 electron carrier along with the iron sulfur centers. You can see the structure of NAD plus and you can see the structure of FAD. So, FAD is derived from vitamin riboflavin and NAD is derived from nicotinamide. When you are talking about the mitochondrial inner membrane, there are 5 complexes found in the mitochondrial inner membrane. The first complex is known as NADH dehydrogenase. The second complex is known as succinate dehydrogenase. The third complex is known as cytochrome BC1 complex. The fourth complex is known as cytochrome oxidase, and the fifth complex is known as ATP synthase. Out of these 5 complexes, the first 4 complexes are responsible for the proton pump and the fifth complex is responsible for the ATP synthesis. So, we have 2 reducing equivalents NADH and FADH2. NADH giving its electron to the complex first. So, the electron transfer take place in complex starting from the complex first when it comes from NADH. Electron transfer that taking place starting from the complex second comes from FADH2. So, we will discuss how NADH giving its electron to the complex first and how this electron coming from NADH is responsible for the proton pump in different complexes and ATP synthesis. We will also discuss how this FADH2 giving its electron to the complex second and what is the path of electron transfer in the different complexes and ATP synthesis starting from FADH2. You can see the 4 electron transfer carriers which is found in the mitochondrial inner membrane. At the same time, you can also see complex 5 which is a proton pump channel. When you are talking about the complex first have FMN as electron carrier, iron sulfur center as electron carrier, and ubiquinone which is a final electron acceptor of this complex. So NADH which is produced in various metabolic pathways are utilized by the complex first. So electron from NADH pass to FMN. So NADH undergoes oxidation and FMN undergoes reduction. Similarly, when FMN gives its electron to iron sulfur center, it get oxidized and iron sulfur center get reduced and electron finally reaches to the ubiquinone one by one. As you know that iron sulfur center is 1 electron carrier, so they will transfer 2 electron of NADH one by one. So we will have finally transfer of 2 electron from NADH to the ubiquinone, and that will have production of reduced ubiquinone. During this process, some proton are pumped. The mechanism of proton pump will be discussed in the next modules. The ubiquinone which is produced in the complex first give its electron to the complex third. So in the complex third we have different electron carriers. 2 electron from ubiquinone is given to the cytochrome C one by one. So first reduce ubiquinone giving its first electron to iron sulfur center, and then iron sulfur center give that electron to cytochrome C1, and then cytochrome C1 give its electron to the cytochrome C. But after giving one electron to iron sulfur center, the semiquinone will be produced. Similarly, semiquinone give its electron to cytochrome Bl, which give its electron to cytochrome BH, and finally cytochrome BH give its electron to the oxidized ubiquinone, which is converted into semiquinone. So net result of electron transfer by one reduced ubiquinone is production of one cytochrome C and one semiquinone. The second reduced ubiquinone give its electron again in the similar fashion to the cytochrome C, but the semiquinone which is produced here giving its electron to the semiquinone via cytochrome BH, cytochrome Bl, and then that semiquinone which is produced is converted into reduced ubiquinone. So net effect is we will use 2 reduced ubiquinone and producing 2 cytochrome C, and we are regenerating 1 reduced ubiquinone. So the net effect is we are going to produce 2 cytochrome C, each one of having 1 electron, and this cycle is known as Q cycle because ubiquinone is recyclized. Then 2 cytochrome C which is produced in the complex third, giving its electron to the complex 4. But during the electron transfer in the complex third, we will also pump proton from matrix to intermembrane space. The 2 cytochrome C produced here giving its electron to the complex 4. So the copper A is the first carrier of the complex 4 which is accepting the electron from cytochrome C. It will transfer it to the copper B via cytochrome A and cytochrome A3, and finally the 2 electron one by one From cytochrome C will reach to the oxygen, and oxygen is converted into H2O. During this process, again the proton pump take place. So when we are starting electron transfer from NADH, complex first pump proton, complex third pump protons, and complex 4 pump proton, and only these 3 complexes are involved in the electron transfer starting from NADH. At the same time, when we are starting electron transfer from succinate or FADH2, then complex second involved, and complex second give its electron to the complex third via ubiquinone, and then complex third give its electron to the complex 4 by cytochrome C. So during this process, starting from FADH2 or succinate, only complex third and complex 4 is going to pump the protons. So when proton pump from matrix to intermembrane space, in the intermembrane space, they all proton are accumulated, and once they are coming back, they are coming via ATP synthase, which is fifth complex, and then during this proton transfer, the ATP synthesis take place in the complex 5. We have discussed how complex first is involved in the electron transfer and what are the different electron carrier of complex first. So during the electron transfer in the complex first, the standard redox potential difference will be created, and that is converted into Gibbs free energy, and that Gibbs free energy helps complex first to pump proton from matrix to intermembrane space. At the same time, they will also produce one reduced ubiquinone. It is very important point to note that premature leakage to oxygen occur in the complex first. So this is a major site for the production of reactive oxygen species. Complex second have FADH2, iron sulfur center, and ubiquinone as electron carrier. The important point of the complex second is that it is not a transmembrane complex, and the redox difference which is created in this complex are low. These two reasons makes this complex unable to pump any proton, but electron transfer in the complex second leads to the production of reduced ubiquinone that is used by complex third. So any ATP that is produced starting from FADH2 is only produced because of involvement of complex third and complex 4. Complex third have a set of electron carrier such as risky iron sulfur center, cytochrome B, cytochrome C1, and cytochrome C. They all are involved in the electron transfer of complex third. Told you, one ubiquinone give its electron to risky iron sulfur center and finally to the cytochrome C via cytochrome C1, and second electron of ubiquinone is transferred to the cytochrome B, which finally produces a semiquinone. And during the utilization of second molecule of reduced ubiquinone, we will produce again one more cytochrome C molecule, and the semiquinone is finally converted into reduced ubiquinone. Therefore, the net result of electron transfer in the complex third will be utilization of two reduced ubiquinone molecule and production of two cytochrome C molecule. The cytochrome C molecule will transfer its electron to complex 4, and during electron transfer in the complex third, the redox potential is created which pumps two proton from matrix to intermembrane space. Electron from complex third reach to the complex 4 via cytochrome C and pass through electron carrier copper A, cytochrome A3 and copper B. The oxygen atom is the last electron acceptor of complex 4 get reduced into H2O, and during the electron transfer, the standard redox potential created in the complex 4 will be responsible for pumping 4 proton from matrix to intermembrane space. Therefore, If electrons start from NADH, then it can pump 10 proton from matrix to intermembrane space by involving complex first, third and fourth, while if electron transfer start from FADH2, it will pump 6 proton by involving complex second, third and fourth. Complex 5. Complex 5 is also known as ATP synthase. It consists of two major subunits, FO, which stands for oligomycin sensitive, and F1. And stoichiometry of this complex is alpha 3, beta 3, gamma, delta, epsilon. These are the part of F1, and FO consists of A, B2, C 9 to 12. C 9 to 12 means the number of units of C varies in different organisms. You can see this structure where you can locate the presence of A subunit, B subunit, C subunit of FO and other remaining alpha, beta, gamma, delta, epsilon of F1. You can see that C subunit and A subunit, they are basically membrane bound subunit, and A subunit linking the B subunit and B subunit linked with the delta subunit. So when proton transfer take place, it always pass between the spaces created between A subunit and C subunit, and movement of this proton is responsible for the function of this complex. As you can see from structure of ATP synthase or FOF1 complex, ATP synthase can be functionally divided into rotator and stator. Rotator consists of C subunit, gamma subunit, epsilon subunit. Stator consists of alpha subunit, beta subunit, delta subunit, A subunit and B subunit. When we are talking about the function, these two independent functional units works together. So the movement of proton from intermembrane space to the matrix via ATP synthase leads to the rotation of rotator in one direction that develops torque into the beta subunit. As you remember, the beta subunit is a part of F1 complex. So the torque developed into the beta subunit is responsible for the ATP synthesis. What is the mechanism of this ATP synthesis? That will be explained by binding chains mechanism. And what is the reason of this proton pump from intermembrane space to back into the matrix will be discussed in the next module. The binding chains mechanism is used to explain ATP synthesis. In this mechanism, torque developed by the movement of rotator will be responsible for conformational change in the ATP synthase. And beta subunit exists in three conformation. You can see in the picture, one conformation is open conformation which doesn't have anything. Other conformation is loose conformation which have ADP plus PI, and third conformation is tight conformation which have ATP. When 3 proton moves from intermembrane space to back into the matrix, the sufficient torque is developed which causes conformation change in the beta subunit. Therefore, if you want a beta subunit to convert from one conformation to other conformation, you have to pump 3 proton from intermembrane space back into the matrix. Therefore, for changing all the 3 conformation, we need 9 protons. So, same 9 proton is utilized for synthesis of 1 ATP by 1 ATP synthase. But if you remember the structure of ATP synthase, there are 3 beta subunits. So, the same 9 proton also utilized for the production of 2 more ATPs. Therefore, we can say that a total of 3 proton are required for synthesis of 1 ATP because 9 proton produces 3 ATPs. 1 additional protons are required for phosphate transporters, and that is mandatory when you want to your ATP synthesis to be continued. Therefore, that will be also included in the number of proton required for one ATP synthesis. Therefore, for all the calculation, we are always considering 4 proton as a required number of proton for synthesis of one ATP. So, student, let's summarize what we have learned from in this module. So, we have learned what are different electron carriers present in the mitochondrial inner membrane. The carrier include complex first, that is NADH dehydrogenase, succinate dehydrogenase as complex second, cytochrome BC1 complex as complex third, cytochrome oxidase as complex 4. So, these 4 complexes are responsible for electron transfer when you are starting from NADH or FADH2. These electron transfer in these complexes are responsible for proton pump from matrix to intermembrane space. In the intermembrane space, the proton created two things. First, they have created electrochemical gradient. The second, they have created proton motive force. This electrochemical gradient and proton motive force together are responsible for backward movement of proton from intermembrane space to the matrix, and movement of proton from intermembrane space to the matrix is responsible for ATP synthesis. This electron transfer in the different complexes creates proton movement, and the proton movement via ATP synthase create ATP synthesis. So, the movement of proton from intermembrane space to the back into the matrix leads to the conformational changes in the ATP synthase, and that conformational changes are responsible for ATP synthesis in the beta subunit of ATP synthase. So, when we are talking about ATP synthesis starting from NADH, so NADH pumps 10 proton from matrix to intermembrane space, including complex first, complex third and complex 4. These 10 proton when come back from intermembrane space to back into the matrix, they will produce 2.5 ATP, because 10 divided by 4, so is going to be 2.5. Similarly, when electrons transport start from FADH2, it will pump 6 proton from from matrix to intermembrane space, and these 6 proton are responsible for 1.5 ATP. Please remember one thing, we can produce either 2 ATPs or 3 ATPs, either 1 ATPs or 2 ATP. So, we will not able to produce 2.5 ATP, but we are saying 2.5 and 1.5 to make it clear about number of proton that is pumped from matrix to intermembrane space and ATP synthesized by ATP synthase. Thank you.